Hello dear students. In the chapter Cell, the unit of life, so far we have studied various aspects of the cell. Let's quickly recapitulate it. We started with the different types of cells. First we studied the structure of a prokaryotic cell, and then we switched over to eukaryotic cell. We differentiated plant cell from animal cell. Then we studied about various parts of the cell, starting with cell wall, followed by cell membrane. Then we switched over to endomembrane system, followed by the mitochondria and the plastics. And in the last lecture, we studied about the nucleus. Now we have come to the last segment of the chapter, where only few structures are remaining and they are worth studying. What are they? They are cytoskeleton, centrosome, cilia and flagella, and ribosomes. So in today's lecture, I will be explaining about these structures. So let's begin with the first structure, which is called as cytoskeleton. So cytoskeleton is an elaborate network of small fibers which may be in the form of microfilaments or microtubules. They are made up of special proteins and they are responsible for providing the mechanical support to the cell. They are also responsible for intracellular transportation within the cell. And the most important, they are responsible for maintaining the cell shape because they act as a framework for the cell. And they are also responsible for cell motility. So, this is the first part, that is cytoskeleton. Let's move to the next part. Now let's study the structure which is present only in an animal cell. This structure is absent in a plant cell. We are talking about centrosome. Centrosome is actually a pair of centrioles which are perpendicular to each other. They are present just near the nucleus of an animal cell. And if we take one centriole and take a TS of it under the electron microscope, it gives a beautiful structure which is called as a cartwheel structure. Why cartwheel? Because if you see, this structure is made up of 9 triplet microtubule bodies which are peripheral. There is no microtubule in the center. Therefore, it is called as 9+0 arrangement. And these microbodies are connected to each other with the help of linkers, and in the center, that is hub, they are connected with the radial spokes. These centrioles are very important structure because they are not only the part of the basal bodies of cilia and flagella, they are also responsible during the cell division process in the formation of spindle fibers. So this is the structure of the centriole. Now let us switch over to cilia and flagella. In the animal cells, the centriole also act as the basal bodies for the structures called cilia and flagella. Anatomically, cilia and flagella are similar. The only difference is cilia are small outgrowths and flagella are long outgrowths. Cilia are generally seen in the bronchi, in the fallopian tubes, whereas the flagella is very commonly seen as the tail of the spermatozoa. Now if you see the cross section of cilia or flagella, we will see that the core of the cilia and flagella is called as axoneme, which is surrounded by a single plasma membrane. And in the core, there are 9 doublet microtubules. And in the center, there are 2 microtubules. That's why it gives an appearance of 9 plus 2 arrangement. These doublet microtubules, they are connected with the help of radial spokes, as well as there are some protein outgrowths. These cilia and flagella are mainly responsible for providing the movement to the cell in case of flagella mainly, and in case of cilia. In case of paramecium, cilia are locomotory organ. In human beings, as I've already mentioned, these cilia are present in our bronchi and in the fallopian tube for propelling the substance forward. So this is about cilia and flagella. Next is ribosomes. Ribosomes are commonly called as pallet particles because George Pallet observed under the electron microscope about certain particles which are without any membrane, and they are dispersed at various places in the cytoplasm, on endoplasmic reticulum, as well as inside the mitochondria and plastids, and he called them as pallet particles. And they are actually made up of combination of RNA and proteins. Later on, it was found out that these pallet particles are ribosomes, and they are of different types. Like you can see, they may be 70S type, generally prokaryotes, mitochondria and plastids have ribosomes which are 70S type. S stands for Swedberg unit or sedimentation coefficient, which means they are made up of 2 types of the particles, which are 50S and 30S. These subparticles, they combine together to form a 70S ribosome. Whereas in eukaryotes, mainly in the cytoplasm, they are 80S type of ribosomes, which are made up of 2 subunits, which are 60S and 40S. These ribosomes, otherwise, they are just dispersed, but only during protein synthesis, mRNA binds these in the form of this structure and protein synthesis begins. So these ribosomes are in a way factories for protein synthesis during the process of protein synthesis in translation, which the details we will study in class 12th. Okay, so these are pallet particles or ribosomes. And finally, there are few structures which are single membrane bound vesicle like structures, which are called as microbodies. Microbodies may be present from time to time in different plant cells as well as animal cells. They are responsible for specific metabolism processes. For example, peroxisomes, glyoxysomes, spherosomes. They are performing special functions because they have special types of enzymes which are regulating a specific metabolism at particular time, and these are called as microbodies. Okay, so with this we have completed the chapter cell, the unit of life, in detail. I hope you have understood all the aspects of the cell. Even if you have any doubt, you may please ask anytime. So go through the chapter in the NCERT, practice all the diagrams and go through various functions each cell organelle or any other structure which is present in the cell they are performing. Thank you very much.